moderator for this webinar. Today's webinar is titled Generating a Vaccinium Pangenome to Unlock Previously Hidden Genetic Variation, and our speaker is Dr. Alan Yocca. Dr. Alan Yocca is recently graduated from Michigan State University, where he worked under Dr. Patrick Edgar to develop the Vaccinium Pangenome. He currently works as a postdoctoral research scientist with Dr. Alex Harkess at the Hudson Alpha Institute for Biotechnology. Alan's research aims to understand plant genome evolution at diverse phylogenetic scale. We are very happy to have Alan give this webinar for us today, and we're very excited about the knowledge that he's going to share as part of the work that he did for our VacCAP project. Before we start, I just want to say that please, everybody, if you can keep your cameras off and your microphone muted during the webinar, we highly appreciate not interrupting the speaker. Also, if you have any questions, please feel free to put them in the chat or the Q&A box, and we will answer them at the end of the webinar. We will also be sending via email a survey at the end of this webinar, and we highly appreciate if you can provide us some feedback on the webinar. And with that, I'm going to stop sharing my screen, and I'm going to let Alan take over. Thank you.
There it is. Thank you for that introduction, and thank you to the organizers of the webinar. I appreciate the opportunity to speak and share my research on the Vaccinium Pangenome. So the title, Generating a Vaccinium Pangenome to Unlock Previously Hidden Genetic Variation. This work I did with Patrick Edgar at Michigan State University, and it is part of the VacCAP consortium. So let's start with a brief introduction on Vaccinium. Vaccinium is a genus in the plant family Ericaceae. The genus specifically has over 450 species. It's has two commercially cultivated crops, or at least high-value commercially cultivated crops: blueberry, which is illustrated in B there, and cranberry, which is illustrated in D. And recently, a review was published about current and soon-to-be released Vaccinium breeding and research studies. Recommend you check that out in horticulture research. So, in plant genomics and in genomics in general, what we're learning in the last couple decades, or even the last few years, is that a single reference genome is not enough to capture the genetic material and genetic diversity within a species. So I don't know if many of you might have seen this tweet by a researcher at UC Davis, Jeff Rossabara, that works on maize, and they found they are assembling a bunch of genomes for maize and teosinte, which is the wild extant progenitor of maize. They found only about 5% of the genome has alignable sequences in all individuals. So, that's less than a few dozen representatives of the same species or wild species. And they found that a very substantial portion of the genome is only found in a few individuals. So looking through the replies, they might think that, oh no, not much is alignable. So, how can we really perform comparative genomics in these lineages? But it seems we're still optimistic that we just really need to assemble everything that's out there. There's a lot of hidden genetic variation sitting that is not captured in a single reference genome. So it's kind of the pitch to go ahead and sequence everything we can, even if it's called the same thing. It might have drastically different genetics. So in comes the concept of a pangenome. So if a single reference individual is not enough, then what happens when we sequence multiple reference individuals? The term pangenome was first coined in this publication in PNAS around 2005 on a study for bacterial isolates. Oops, can I go back? There we go. The phrase where the term pangenome was conceived was they sequenced a bunch of different genomes and suggest that bacterial species can be described by its pangenome, pan from the Greek word meaning whole, and it includes a core genome that contains genes present in all strains, every single individual, and a dispensable genome composed of genes absent in at least one or more strains that are unique to each strain. So to kind of illustrate this graphically, on the top line, I'm showing a schematic of the reference genome for cultivar 1. You have 3 genes in a row, gene 1, gene 2, and gene 3. If we sequence cultivar number 2, we find in the same region, gene 2 can be completely absent. In cultivar 3, we see gene 4 is present in cultivar 3, but not present in our reference. And within cultivars, genes can have different copy numbers, and even variable or even dispensable genes can have different copy numbers. So plant pangenomes recently are available for dozens of species. So I made this list about probably 3 or 4 months ago, and I'm sure it's already out of date. There are many sequencing efforts underway to develop pangenomes for plants, and Vaccinium now makes the list. So beyond the pangenome concept, if you think of a pangenome as multiple genomes within a single species, there was a recent opinion piece that came out talking about a super pangenome that could be developed by integrating the wild side of a species or integrating sort of different species within the same genus. So that's kind of the approach we took for Vaccinium, where we sequenced within Vaccinium corymbosum, blueberry, and Vaccinium macrocarpon, cranberry, to develop our sort of super pangenome. So we keep taking things a step further. So I mentioned earlier that a pangenome contains core and dispensable genes. Recently, there's some thought that dispensable is not the best term. So throughout the rest of the talk, I'll be using the term variable. So just because a gene is absent doesn't mean it's not necessary. So dispensable might be a bit of a misnomer. Across plant pangenome studies, there are a lot of consistencies for characteristics of core genes, or those genes found in every single individual. So core genes are often enriched for essential gene processes such as metabolism or photosynthesis. They are generally longer than variable genes, and they're also more likely to be, found in distantly related species. Variable genes, on the other hand, they're present in a few or sometimes even a single genotype. They're enriched for potentially high-value processes such as disease resistance or any sort of stress resistance or phenotypic differences between genotypes. They're generally shorter than core genes, and they are less likely to be found in distantly related species. So our group decided to apply the super pangenome concept to Vaccinium. The plan was to sequence a bunch of blueberries from northern highbush and southern highbush, and a bunch of cranberry genomes, and through extensive comparative genomics, kind of determine what genes are core, what genes are variable, and just sort of generate this great resource for Vaccinium that was previously unavailable. I'm going to split the talk into 2 parts. I'm going to first talk about the cranberry pangenome side, or the Vaccinium macrocarpon pangenome, and then I'll transition to the blueberry or Vaccinium corymbosum pangenome. So for cranberry accession selection, work done previously with, I believe, short sequence repeat markers, identified some genotypically diverse cranberry accessions, and we selected 10 individuals shown on this slide, plus the reference cultivar Stevens. We didn't sequence it here, but we had that genome available to us, and we de novo / reference guided assembled individual genomes for each of these accessions. So we selected our 10 individuals. We collected leaf and berry tissue, sequenced genomic and RNA sequencing information, used a hybrid de novo reference-guided assembly method to generate genomes and gene annotations for every single cranberry individual. We use this program called Progressive Cactus to align all 11 genomes against each other, and we use the alignment information in conjunction with OrthoFinder 2 to identify core and variable genes in every single genome, which is really incredible. Also, for each genome, transposable elements were annotated. This is done by a graduate student in Patrick's lab, Scott Teresi, using EDTA version 2. The analysis is private on GitHub right now, but I believe in the next few months it'll be publicly available. So this is kind of a big table with a lot of numbers. The main takeaway I want to show here is that a high percentage of the genome consists of transposable elements. There are diverse types of transposable elements, and these show different abundances. So some transposable element classes could have about 9% of the entire genome be that specific class, whereas other transposable elements can be a very small percentage of the genome. Also, this last column would be the standard deviation across all of our cranberry genomes. So there is a little bit of variation in the abundance of different transposable element classes across individuals. Here I'm showing a bar plot of core and variable gene counts. So core in pink on the top, variable in gray on the bottom. So for each individual, you can see the number of core genes in the genome is pretty consistent across individuals, yet the number of variable genes within each genome is much more dynamic. So we have thousands and thousands of both core and variable genes in these genomes, which this is a pretty cool result. Here we're modeling the size of kind of the core and variable genome. It's a bit much to kind of take in here, but basically along the x-axis we have the number of genomes. Along the y-axis is the number of genes. We've got two different lines. So just consider the pink line here, where as we increase the number of genomes, the number of core genes is decreasing. So the more you look at different genomes, kind of the less and less core genes you find. There will be more genes that are just missing in some individuals, and we expect that to plateau at a certain point at the true number of core genes. So this line plateaus fairly sharply at the end. So this just tells us that, you know, the more we sequence genomes, we're probably not going to decrease this core count much. In other words, we've captured nearly all of the core genes present in Vaccinium macrocarpon. Now, considering the gray line, it shows the number of variable genes as we increase the number of genomes. So that is not as close to a plateau, which means there still might be some variable genes within Vaccinium macrocarpon we've yet to sample, but that's just that's a pretty exciting result in that there's still more out there in cranberry germplasm that we could find. But we've still done a pretty good job. We found you know, a lot of variable genes. So we looked at differences between core and variable genes in a few different metrics, just general metrics. So these are all distributions of all core and all variable genes. For gene length, we see the distribution of core gene lengths is much longer than the distribution of variable gene lengths. So this matches up with previous findings in plant pangenomes that core genes are very often longer than variable genes. We break that down by, so this would be transcription start to transcription end site. This second graph is CDS length, so how much protein coding sequence length differences are there. Still, core genes are longer. We find core genes have longer introns than variable genes and also have more introns than variable genes. So by proxy, exon count shows a longer distribution for core genes than variable genes, which is a great result, very consistent with what we find in a lot of other species. Go enrichment, or gene ontology enrichment for core and variable genes. Again, consistent with previous studies, we find core genes are enriched for processes such as plant type cell wall organization, cell wall biogenesis, regulation of cell cycle, these sort of core processes that are necessary in every single individual. Whereas our variable genes have this kind of cool pool of functions, and they're enriched for defense response, regulation of DNA methylation, defense response to fungus, things that might be different between different Vaccinium macrocarbon accessions. So that kind of wraps up the Cranberry portion, and now let's move on to the blueberry reference genome. So for the blueberry reference genome, the blueberry Pen Genome. The blueberry reference genome was published in 2019. It is a haplotype phased genome. So what does that mean? Is it means we have a full assembly for sort of each haplotype. These are our blueberries that we selected. For this portion of the talk, I'm going to be talking about northern high bush. So these are all northern high bush accessions. They were selected with the same method as sort of this low pass genotyping principal component analysis to identify genetically diverse accessions. And blueberries are polyploid. So this is sort of the crux of the reason why I wanted to separate Vaccinium macrocarbon and Vaccinium corymbosum pangenomes. It's because our methods of identifying core and variable genes are slightly different because blueberries are polyploid. So the Vaccinium corymbosum that we're working with is tetraploid, which means it has 4 total copies of each genome. And sort of here is a figure from the blueberry reference genome paper. The base chromosome number is 12, and you see we have 48 assembled chromosomes that represent each sort of what we call a subgenome of blueberry. So what gets a little murky in polyploids when it comes to pangenomes is kind of identifying what is a core and what is a variable gene. It's not really a straightforward question, because you could have, with a whole genome alignment, you could align, let's say, chromosome 1 from individual A with chromosome 25 of individual B, which specifically which chromosome is homologous to which in a genome alignment is not straightforward. And also what happens when there are tandem duplications only on select subgenomes. So we could have a gene that is present in all 4 haplotypes, is what we call these, or we could have a gene that's in a second individual missing on only one of the 3 haplotypes. So sort of how do you determine if that's really core or variable? Is the gene still functional if it's present in 2 copies versus 4? So it got very murky. However, we kind of looked out of it. W85 to the rescue. So we have Vaccinium genotype called W85 that had their genome recently published in this publication, that it is a diploid it's considered sort of a diploid representative of Vaccinium corymbosum. We have a nice phased long read assembly. We phased again, meaning we have a different assembly for each haplotype, and it's a diploid, so we have 2 haplotypes there. It's about a 600 megabase genome and about 35,000 genes per haplotype, which is consistent with Vaccinium corymbosum. So using a diploid reference, by aligning our polyploid assemblies to the diploid, we could identify whether the diploid gene is present or absent in each Vaccinium accession. So that does give us a few limitations, in that now this Vaccinium corymbosum pangenome is reference-based, whereas for Cranberry we had core or variable for each gene and each accession. But this really helps us kind of tease out difficulties with polyploidy in Vaccinium corymbosum. So this is really great. I'm showing you a similar bar plot as for Cranberry, in that we see across accessions the number of orthologs it shares with W85. So we can only call the W85 copy present or absent for each genotype. Therefore, we can't generate the variable gene count as easily. So we'll be just focusing on W85 reference things, and we see, as in Cranberry, the number of core genes is pretty consistent across individuals, but there is a little bit of variation. So our W85 genome, though, is phased, so we can break this down by haplotype. In pink, we have the haplotype 0, or the first one, and in purple, we have haplotype 1. And we see there are, maybe a few less core genes on haplotype 1 compared to haplotype 2. So that's kind of cool. There might be a little bit of variation in what's conserved across different, maybe, parental lineages. So we also have transposable element annotations for each Vaccinium corymbosum genome, which is awesome. Thank you to Scott Teresi again for performing that. The table, pretty similar takeaway messages in that a high percentage of the genome is transposable element. Different classes of transposable elements have different abundances. One thing to notice compared to the last table here, it's hard to tell because I don't have the table on the same screen, but the standard deviations are much lower. So it seems that transposable element abundances are a little more consistent in Vaccinium corymbosum compared to Vaccinium macrocarpon. So we have a similar plot to earlier, showing modeling of the Vaccinium corymbosum core genome. So I also split it by haplotype here, haplotype 0 and haplotype 1. This is kind of giving an indication that the core genome does not decrease too much as you add accessions, but we are plateauing, so we have probably captured most of the core genes within Vaccinium corymbosum. I'm going to go ahead and zoom in on this same plot just so we can see the differentiation between haplotypes a little better. And this result will kind of show us something similar to the bar plots, where haplotype 0 has a few more core genes than haplotype 1. So let's check gene statistic differences, as we did earlier. In pink are core genes, in gray are variable genes. Not as striking as Vaccinium macrocarpon, but we do see that core genes are longer, core genes have longer introns, and core genes also have longer coding sequences, though the difference is not quite as striking. This table is an analysis that Pat did. It was checking the number of core genes shared outside of Vaccinium corymbosum. So earlier I mentioned plant Pen Genome studies found core genes were more likely to be found in like kind of more distant species than variable genes. And we were curious, as we go further out from Vaccinium corymbosum, how many sort of genes are shared. So Pat split it up into two classes, hardcore and softcore. So it's just a little more stricter definition of core. I like to use the word core to mean present in every single individual, but some people make a class called softcore, where it's allowed to be absent in maybe one or only a few individuals. So whereas hardcore would need to be present in every single individual. So we have a higher number of hardcore genes found in these sort of sister Vaccinium species compared to softcore, which is expected because softcore are a little more variable at the species level. But though about, what was it, 50 to 60% of all Vaccinium corymbosum genes are core, when you look compared to further away species, a lot, like 75, 80% of these core genes are found in sister species. So that if it was random, it'd be about 50%. So we're seeing that indeed core genes are retained at a higher rate in sort of sister species. So possible mechanism of gene birth and death, why are there core and why are there variable genes? Where did they come from? If a variable gene is only found in a single accession, how did it get there? That's a super fascinating question. It's as in everything in science, it's not totally worked out yet, but we have some ideas. Mainly gene duplication. So you could have a gene that tandemly duplicates, and then over time, one copy might accumulate certain mutations so that it's distinct enough from the original gene duplicate, but now it has a new function. There could also be an involvement of transposable elements. So a transposable element could, maybe carry a gene or even part of a gene from one location in the genome to another. I think that the mechanism of kind of gene birth and death and where variable genes come from is a fascinating question, and I think work determining those mechanisms is on the horizon. A few future directions of this work. A key one would be improving efficiency of genotyping arrays. So for Vaccinium corymbosum, Vaccinium carpon, now that we know which genes or which regions of the genome are conserved across all these sort of accessions, we can develop genotyping arrays to be able to capture sequence in a lot of many accessions that maybe we haven't sequenced yet, and we don't know completely what's in there. And what's also cool is, let's go back. I'm going to go back real quick to this table showing that there are thousands of genes conserved between certain Vaccinium species and Vaccinium corymbosum. Therefore, we could develop genotyping arrays that can capture regions of the genome we know are present in these other Vaccinium species. So if we ever wanted to integrate wild germplasm into Vaccinium, we kind of have a better idea of the regions we can target thanks to this pangenome study. We could also possibly do a GWAS or QTL reanalysis. There have been many publications out recently that have done sort of some GWAS reanalysis within a pangenome framework, and find they do get localization hits to these variable regions, or these regions that you did not know were there, that were not in the single reference genome. So that'll be exciting. We'd also like to extend this pangenome framework to southern highbush. Here, I didn't do so because of the polyploidy issue, and we were a little bit reference limited, but we'll hopefully work on southern highbush soon. And we'd also just like to use this as a resource available to the community, and really kind of encourage people to dig in and take it for their own applications. They're available on the VacCAP website. I believe all the northern genomes, for sure, I checked. I didn't check the Cranberry, but I believe they're also available. So that have many people to acknowledge, many people helping with this work. I'd like to encourage you to visit the VacCAP website, vacciniumcap.org. They have some fantastic intro slides available on what the project is, and what sort of all of our goals are. I really appreciate all the hosts of the webinar and the organizers for giving me this opportunity to kind of speak and chat about my research, and I'd like to take any questions.
Thank you so much, Alan, for that wonderful webinar. Any questions? I don't see any questions on the chat, but I think that the audience is not that big, that if anybody wanted to unmute themselves and ask a question directly to Alan, they could go ahead and do so. I see one question here from Felipe. It says, great talk. How do you envision using the Pen genome from downstream analysis, for example, GWAS? Should we redefine the classical nomenclature of reference and alternative alleles for future genomic analysis, as we will have multiple genomes as part of our genome of reference?
Yeah, it's a great question. I appreciate it. So for sort of using Pen Genome for GWAS reanalysis and the reference alternate question, I think pretty soon, instead of representing the genome of a species as a linear, single reference, we'll move more towards the graph Pen Genome representation. I wish I would have talked about that a little here because it's fascinating. Whereas instead of a linear graph of a reference genome, you can have little bubbles and little sort of branching paths that represent what sort of different sequences could be at the same loci. So I think in future genomic analyses, you could maybe go to the graph Pen Genome or the graph genome representation, and I know there are some tools available for GWAS specifically on graph-based genomes.
Great. We have another question from Paul. How important is proper phasing of an assembly to getting the pangenomics right?
Yeah, so I guess there's no right way to do pangenomics, but certainly phasing can be important, but I think it depends on the species. So here for Vaccinium, we found phasing the W85 genome, there were slight differences, but maybe only a couple hundred gene differences between haplotype 1 and haplotype 2. I know for apple and for grape, they're very hemizygous. So that means there's a lot of haplotype variation. There could be large sections of deletions on one haplotype in reference to the other. So I think for some species, phasing will be very important. And also, I think phasing will be very commonplace relatively soon because of improvements in sequencing technology.
Okay, so I have a question from Lily. Says, will the pangenome be available for others to use? If so, where?
Yes, thank you, Amaya, for responding in the chat. They are publicly available. We have genomes, we have annotations, we have protein FASTA files available on the VacCAP website, vacciniumcap.org. This is the publication is forthcoming. It's one of those, last chapter of my PhD, and now I've started a postdoc, so I'm trying to work on it on the evenings, and it's this fall, I anticipate it at least being a preprint, but the data is publicly available. Please go to it, jump on it, use it if you need it. And I'm available. My contact information I will put in the chat if you have any questions about the data.
Great. Okay, so we have a lot of other questions. So Gregory is asking, for the W85, how did you decide which copy of each chromosome was, you're gonna have to help me here,
Hap0
and
Hap1. I'll have to double check with the published genome. I was not directly involved in that. I believe it was based on a linkage map. I think that's how they assigned it. But it also sometimes in these assemblies it's relatively random. So one homologous copy for each chromosome will be randomly assigned. We are able to phase on a single chromosome specifically what is on Hap0, what is on Hap1, but we cannot say is Hap1 from chromosome 1, Hap1 from chromosome 2. That might get shuffled, but I'd have to double check the publication for that. I'm sorry.
Good. So then there's Juliana asked, are you going to make the pangenome graph available too?
I did not generate a pangenome graph for this. Sorry. I think the data is available to generate that if you'd like to do it.
Okay, so Felipe has a second question is, does it make sense to combine southern high-bush and northern high-bush in the same pangenome framework?
So it could. I think the exclusion of southern here was just a result of timing. So initially we wanted to generate the most accurate genome for each individual. So I mentioned our genome assembly was hybrid, de novo, and reference based. So we had a southern high-bush reference for our southern high-bush genomes. Now that was tetraploid, so that was that had our, our multi-copy, what's a core, what's a variable genome call. So those were not as easy to align to the W85 genome. I think it could make sense. They're definitely they're pretty close genetically, but for this specific project, I think it was just a victim of timing of when we decided what method we were going to use to call core and variable. One quick interjection is I've been told that the haplotype 0 and haplotype 1, which chromosome is in which, was determined by just the longest assembled scaffold for that. So they are kind of switched across chromosomes.
Perfect. Jim Polishuk wants to ask, he has two questions and he's going to unmute himself and ask.
Okay.
Go ahead, Jim.
Okay, so the first question is, how do you, at what point, how variable does it have to be to call two different sequences, two different genes?
Good question. It depends, like everything, right? So what helps in this framework is we used syntenic information. So we called a gene core if it both aligned at a high percentage and it aligned at the right place. So we were able to kind of look around a gene and see what genes were kind of nearby. So in that case, there could be something with like a relatively high similarity, but if it wasn't in the right place, then we didn't call it core. Whereas for how low does the similarity have to be if it's in the right place, kind of an arbitrary threshold. I think we chose like 70% identity.
Okay, so my next question is, most northern highbush varieties are interspecific hybrids, and southern highbush varieties are also interspecific hybrids, mostly Vaccinium corymbosum, Vaccinium darrowi, and some Vaccinium angustifolium. So it seems to me that this kind of analysis gets really complicated, but I'm also wondering if, since you have the Vaccinium darrowi, a Vaccinium darrowi reference, right?
Mm-hmm.
And you have a highbush corymbosum reference. When you look at the synteny between them, do you find that some genes or gene sections sort of drop out by species? You expect there to be variability, but you also expect things to sort of pop out, like Vaccinium darrowi should have a whole bunch of things that corymbosum doesn't have, and vice versa. Yeah,
I haven't checked a synteny plot between those two specifically. I think Patrick might have, so it'd be worth reaching out to him about it. I could see definitely sections of corymbosum that are more similar to just specific sections of darrowi. What I'd also be interested in which specific haplotypes in the corymbosum reference might associate. Is it the primary haplotype, or is it across all four? So yeah, that'd be interesting to check out.
Oh, one more quick thing before I lose the floor. We also have to remember that blueberry cultivars are very highly selected. And I wonder how high selection affects your results. W8520 is a wild collection from New Jersey, but most of the other ones are very highly selected. And it seems to me that high selection pressure would skew the core and variable genes that you find.
Yeah. I think so, but yeah, so I guess there are a lot in corymbosum that is probably not in W85 that have been selected out, right? Is that?
Yep.
Yeah. So yeah, hopefully hopefully the sort of variable regions that we identified in corymbosum would be contributing to some of those selected traits.
Okay.
All right. We have a lot of other questions in the chat, and I think that you might have referred to this already, but now it's asking, why did you separate the northern highbush from the southern highbush for the blueberry pangenome, since the majority of the southern highbush is also a Vaccinium corymbosum?
Yeah, so initially, again, I think it was just a victim of the timing for us. So all our southern high bush genomes are based on a different reference than our northern high bush genomes. So comparing the northern to the southern high bush across each other, you have 4 haplotypes against 4 haplotypes, and things got a little murky. We could have easily just we could as easily aligned all the southerns to W85 and then done a southern specific pangenome, but we couldn't really integrate the two together. Comparing them would have been a bit murky, and I think that is something we're working towards. Maybe somebody else in the Edger lab is doing that, but I kind of got the clean cut at this stage of the project.
Perfect. Okay, so Fernando is asking, can you elaborate a bit on what kind of genes are part of the core genes across Vaccinium? Are there more paralog than ortholog genes?
So the identifying the genes present in other species. So, I showed in Corymbosum, you have about like 20,000 genes per haplotype that are shared across every individual, whereas for the, let's say, DARY, there were 11,000. So there are definitely less genes that are core when you compare it to an outgroup. These are also pretty much all going to be orthologs. I don't think we looked at paralogs, but so paralog would be sort of an intra-genomic duplication, having more than one copy in a single individual. We know that variable genes show higher rates of tandem duplication than core genes, so there were probably more orthologs than paralogs for core genes. And also, the further out you go phylogenetically, the more kind of core processes that these genes get associated with. So, if you go far enough out, it's kind of just like what makes a plant a plant.
I love to see all these questions. A lot of interest in everything that you presented. So the next question is from Joe. He's asking, when determining core versus variable gene sets, have you identified any alternate genetic pathway separate from gene duplication where core or essential genes do not necessarily need to be present within the genome, provided there is a viable alternate?
Yep. So that's a key reason why I like to use the word variable and not dispensable. A lot of people still use dispensable, but yeah, so you could have 2 genes that don't look anything alike that perform a very similar function. So maybe like half your individuals have gene A, half have gene B, but they get the same thing done. And in my pangenome framework, the way I do things, I'm going to call that a variable gene, even though it is going to be a core regulator or some sort of like core function. I think that's just kind of I think that's an important limitation of pangenome studies, and an important reason why I like to show distributions of effects across the 2 classes, because there are definitely variable genes that have core essential cellular processes. There are definitely core genes that have maybe very not essential, and you could cut it out of blueberry and nothing would go wrong. So that is an interesting thing to keep in mind.
Perfect. There's a comment here from Juliana about her previous question about the graph, and she says that she thinks it would be interesting for the community to use the same pangenome graph so we can exchange markers, QTLs more easily. It's a comment. Then Fernando, I think, has a follow-up to his first questions of, do you think we're lacking in the ontology of functional genetic studies to be able to say more about whether genes are paralogs or orthologs?
I guess I'm not too sure on the question. So determining whether it's a paralog or an ortholog, do you mean like on the same haplotype? Maybe I'm forgetting the terminology here. But for functional,
sorry about that. I don't know if you want me to comment on that, but
yes, please.
Yeah, it's essentially, I was just wondering, when you run your genomic study and you start, subsetting all the genes into core and and the other groups that you have, I always wonder, you're talking about synteny and how, like, you know, you're using synteny to determine the core genes. And I always wonder, you know, with how much information do we have, you know, from like the gene ontology to be able to say whether between accessions, the same gene, which is synthetic, is doing kind of a different thing in another cultivar or whatnot. You know what I'm saying there?
Yeah. So like I'm still not getting the parology, but for that specific one, it is whether gene A in cultivar 1 is synthetic with gene A in cultivar 2, whether that gene is doing different things, even though it's synthetic, right?
Yes, exactly. That's so. I didn't get the terminology that well either. So I'm glad you figured it out for me.
Yeah. So it's great. We're good. So yeah. Who knows? Well, I think functional validation of genes is very difficult, and it requires a lot of work. And so I'm always appreciative of those who've done it. I'm not an expert in it per se. You know, I know that you could have a single nucleotide substitution that affects the function of a protein. So I don't know how you're going to identify that for me sitting on my keyboard typing A, T, C's, and G's, but I think the power here would be to look for these broad patterns. So I think the we're just going to try to call core genes using what we believe the behavior of most core genes is like.
All right, thanks, Alan. Yeah, great talk. Thank you.
Thank you.
We have one last question in the chat from Andrew, asking, are introns considered at all in defining core versus variable genes? In other words, if two core genes have less than 70% identity, but the variation is mostly in introns presence, absence, etc., how is this factored?
Great question. Our identity thresholds were strictly CDS, so the protein coding regions. So introns differential presence absence would affect the alignability of the region. So if the intron set was super variable and the flanking regions were very variable, we might lose core genes that have matching CDS, but just really variable introns. But I think our approach captured I guess intron variability didn't play too much noise into our core variable calls. If we had a 70% CDS alignment, I think they mostly align pretty well.
Okay, I lied. There's two more questions now.
That's okay.
We're gonna continue since we still have time.
We have time.
Okay, so Juliana, again, she has another question. Is that, do you know any other studies where they were able to achieve the plateau of core genes you mentioned? And how many individual genomes were necessary to get there?
This is a fun talking point. Great question. So yes, there are studies that show a core in the plateau of variable genes and core genes. Both of them plateau very nicely. And I can't remember specifically which study, which plant study off the top of my head, but they basically plateaued over like 25 individuals, but they sequenced like 300. So a lot of sequencing for not too much more gain. But I say the word 25 to plateau, completely random. As far as like very, very species specific. What you need for the plateau is what are the factors affecting the plateau, not only how many individuals you sequence, but the diversity of the individuals and how much genetic variation exists within that species. I think it seems to me, based on the numbers, Vaccinium has a little bit less genomic diversity than some other species such as Brachypodium. That might be because of domestication in Vaccinium. But yeah, a lot of people ask, how many individuals do I need? How many do I need to sequence for my Pen genome? It really depends, and it depends on your individual selection strategy. So,
great. And the next question is from Janine. Did you analyze whether any of the core or variable genes were the same between the Pen genomes?
So I think you mean between the Vaccinium macrocarpon pangenome and the Vaccinium corymbosum pangenome. We have not been able to draw direct comparisons between the pangenome studies as a whole. We could definitely compare our two references, W85 to the Stevens macrocarpon reference, and compare based on that. I think that's all the further we're going to take it. But it gets really murky with, you have different copy number of certain genes, and they could be, which chromosome is homologous to which chromosome. So that'd be interesting to look at for sure.
Great. I think that those are all the questions that I can see on the chat. I'll just send again the link for our YouTube channel for the Vaccinium CAP. This recording will be posted there, and you can also go and see our previous webinars. I also want to share with you that we will have a survey that is going to be sent your way once this webinar is over. And so please, if you can provide us some feedback, that would be greatly appreciated. We want to really thank Dr. Alan Yocca for a wonderful webinar, a lot of great information, and there was a lot of questions. I love that there's like this very active discussion about the data that you presented and the results you presented. And with that, thank you so much, Alan, for everything. And we look forward to our next webinar next year. And if you want to, again, look again or review this webinar, it will be posted on our YouTube channel. Thank you.
Thank you, everyone.